Hello, everyone. My name is Matthew Lay, and I'm currently an undergraduate biology student at the University of Nevada, Las Vegas. I'm researching under the Department of Chemistry and Biochemistry and under the supervision of Dr. Hei-Soo Khan. Today's talk will be about the synthesis of novel fluorescent probes for the early diagnosis of Alzheimer's disease. To begin with, AD is a neurodegenerative disease that manifests itself through a progressive decline in motor function, memory, as well as cognition. The reason why AD is an area of focus is because it accounts for 60 to 70% of all progressive cognitive impairments within the elderly population. And as the average age of the world's population continues to increase, we will likely see AD at a more prevalent rate in the future. With that said, AD is closely associated with the accumulation of amyloid beta proteins that forms insoluble plaques in the brain. The formation of amyloid beta proteins and insoluble plaques is a hallmark of AD and thus can serve as a means for a diagnosis. Current diagnosing methods for AD consist of MRI scans, CT scans, as well as radioactive material that binds to the proteins at hand. While they are decently effective to diagnose AD, they are quite impractical in the real-world sense. They are not affordable, and they are quite harmful when exposed too long to the patient. And therefore, my primary focus was to synthesize a series of potential fluorescent probe candidates for a practical and safe means to detect AD in its early stages through amyloid beta protein binding as well as insoluble plaque binding. To confirm the identity of the products, I used proton as well as carbon nuclear magnetic resonance, or NMR, and elemental analysis, also called EA, to analyze the product. I further characterized the product through determining its melting point, thermal stability, and fluorescent properties through DSC, TGA, and fluorescence spectroscopies, respectively. As for the methodology, it is split among two steps, as well as two schemes right here. The first step consists of synthesizing the gamma picolinium bromide salts. I focus on 3 different series with differing carbon chain lengths, 6 carbon, 9 carbon, as well as 12 carbons. This starting material will be used to carry on to our second reaction that will occur via Nova-Nagel condensation. The percent under the methodology heading are the percent yields for each reaction. After synthesizing the bromide salt, I further synthesized I participated in the counter ion exchanges to tosylate as well as trifluoride to form our entire series. In figure 1 and figure 2, these are the proton NMR spectrums for C9 tosylate as well as C9 trifluoride, which will be our focus for this poster. The proton NMRs have peaks that corresponds to the protons on the molecule itself, depending on their positions on the molecule. The peaks will be differing in chemical shifts, and thus we can identify the molecule through proton NMRs, and both of these NMRs confirm the identity of our C9 tosylate product as well as our C9 trifluoride product. In figure number 3 and figure number 4, we have the serial dilutions in methanol at a concentration of 10 to the negative 5 of both C9 tosylate as well as C9 trifluoride. We can see that they emit a blue-greenish light under a UV light when dissolved in methanol. On the right side, we have the solid state of both of the compounds, and it is once again under the UV light, it fluoresces as a yellow color, which is different when it's in a solution. For figure number 5 and figure number 6, we have the emission excitation spectra of the C9 tosylate as well as C9 trifluoride in methanol. The main area of focus would be right here as well as right here, which represents the greatest intensity that is emitted from the compound at a specific wavelength. The specific wavelength for the tosylate salt would be 482 nanometers, whereas in the trifluoride salt, it is 484 nanometers. These wavelengths of light corresponds to the color bluish green on a color spectrum. And therefore, we can see that an application when shining the UV light on the solution. For the conclusion, we synthesize C6, C9, as well as C12 parastyropyridinium salts that contain the bromide, tosylate, and trifluoride counterions. The identities were all confirmed through proton, carbon, and carbon NMR, as well as elemental analysis. The UV vis spectra, which was not shown on this poster, showed that both the C9 tosylate and C9 trifluoride compound showed a lambda max peak at 383 nanometers and 384 nanometers in methanol, respectively. What this shows is the greatest wavelength of light at which the compound can absorb. Similarly, or conversely, the emission spectra, which shown right here, shows the wavelength of light at which the compound emits at the greatest intensity, which happens to be the bluish greenish color that we see. Lastly, I carried out solubility tests for all three series of these salts, and I found that they were soluble in DMSO, which was great because DMSO is typically used in experimentally testing if it is compatible and biologically safe for use in vivo or in the human body. For future work, I will further identify fluorescent measurements in different solvents, make further counter ion exchanges, change the carbon chain length of each of the molecules. I can do C14, C1, C3, and so forth, as opposed to the C6, C9, and C12 that I focused on. We can change the structural modifications. We focus on the para position for this molecule right here, but we can also change it to the meta and ortho position on the aromatic ring. Lastly, of course, we would do biological testing in vivo for the fluorescent probes to see if they are binding to the amyloid beta proteins and insoluble plaques in the body. And during the biological testing, we can see once the compound binds to the target protein, it will change its fluorescence characteristic. It could either increase in fluorescence, it can decrease in fluorescence, and that would signify that this compound has successfully binded to the target protein. And that is how we could diagnose Alzheimer's disease at an early age. As for acknowledgements, I would like to thank the UNLV of Undergraduate Research unit for the 2021 Undergraduate Research Stimulus Program, as well as Dr. Bomick, Ronald, Cy, and as well as David for their support in making this project possible. And on the bottom right-hand corner, I have my references. Thank you for listening to my talk today, and I am free to answer any questions that you may have afterwards. Thank you.